Hello, my name is Samantha, and the genetic disorder I decided to do for this project was congenital adrenal hyperplasia. Congenital adrenal hyperplasia is an autosomal recessive disorder and is one of the most common inherited diseases. This disease involves a specific grouping of genetic disorders which cause the adrenal glands to be affected. The adrenal glands are responsible for making hormones such as mineralocorticoids that regulate potassium and sodium levels in the body, androgens like testosterone that are classified as male sex hormones that are needed for the development and growth of females and males, and cortisol, which is what regulates the human body's response to stress or illnesses. People who have CAH have mutation present in their genes that results in deficiency of a specific enzyme that is required in order to make these hormones. For this project, I decided to focus on the most common type of congenital adrenal hyperplasia, which is 21 hydroxylase deficiency type. 95% of all cases of CAH is this type. I decided to split up the characteristics into men and women. For men, one major characteristic is they have growth spurts that are early in life. However, their overall height tends to be shorter than their other family members. Also, there's a good chance that men with this type will experience a decrease in fertility. In addition, they will have small testes. For females, they also will have a stunted growth, and could have an abundance of body hair, which is known as hirsutism. Their menstruation can be irregular, and they could experience male pattern baldness. They also tend to have ambiguous external genitalia that is not clearly female or male, and have a decrease in fertility. For molecular cause, congenital adrenal hypoplasia 21 hydroxylase deficiency is caused by a mutation in the CYP21A2 gene. This part of the cytochrome P450 family of enzymes, this gene is located on the RCCX module, which is a region in the chromosome that is prone to recombination, which can result in rearrangements such as gross deletions, gene conversions, and gene duplications. CYP21A2 gene makes instructions for making the enzyme 21 hydroxylase. When there is not a sufficient amount of 21 hydroxylase enzyme, the substances that naturally form aldosterone and cortisol are built up in the adrenal glands and make androgens instead. This excess amount of androgens lead to sexual development abnormalities. The lessening of aldosterone also contributes to solid loss within the body. Congenital adrenal hyperplasia is inherited in an autosomal recessive pattern. This means that both copies of the gene in each cell have mutations. The parents of offspring with congenital adrenal hyperplasia can each carry one copy of the gene that is mutated but might not show any symptoms. The parents in this case would be called carriers because they each carry a single copy of the mutated gene, which they are able to pass to their child. In order to better describe the mode of inheritance of this particular disease, I have made some Punnett squares, and we'll walk you through them. So in my first Punnett square, it is of both parents are afflicted with CAH, so big C is dominant allele, little c is recessive allele. After completing the Punnett square, the possible genotypes for the offspring in this case could only be little c, or recessive, because both parents only possess the genotype little c. This means that there is a 100% chance of these parents' offspring having congenital adrenal hyperplasia. Also, this means that the offspring will share the same phenotypes as the parents, which are salt-wasting and possibly ambiguous genitalia. For my second Punnett square, it is one parent has CAH, one parent is not a carrier and does not have CAH. So after completing this Punnett square, the possible genotypes for the offspring in this case could only be big C, little c, because both parents are homozygous but of different alleles. This means that there is a 0% chance of these parents' offspring having congenital adrenal hyperplasia, but all of them would be carriers of the disease. Also, this means that the offspring will share the same phenotype of the second parent, which are non-salt wasting and non-ambiguous genitalia. My third Punnett square is one parent has CAH, one parent is a carrier. After completing the Punnett square, the possible genotypes for the offspring in this case could be big C, or little c, because one parent has the disorder while the other parent has one mutated allele and is a carrier. This means that there is a 50% chance of these parents' offspring having congenital adrenal hyperplasia and 50% chance that they might not have CAH, but will be carriers. My fourth Punnett square is neither parent has CAH nor is a carrier. After completing the Punnett square, the possible genotypes for the offspring in this case could only be big C because both parents only possess the genotype big C. This means that there is a 0% chance of these offspring having congenital adrenal hyperplasia. Also, this means that the offspring will share the same phenotypes of the parents, which are non-salt wasting and non-ambiguous genitalia. In this slide, I show you my simple pedigree, which is basically a family tree, showing how different family members may be a carrier of CAH, may have CAH, or may not be a carrier nor have CAH, and how it works through the family tree. It's very similar to the Punnett squares that I just showed you, but it's a more laid out, more in depth through generations version of that. But again, it's much like the Punnett squares. As I zoom in, you can see that as well. So, genetic testing options. So, in prenatal testing, there is chorionic villus sampling. This is where cells are withdrawn from the infant's placenta and looked at under a microscope. And amniocentesis, which is where a needle is placed into the amniotic sac and amniotic fluid is withdrawn and then tested upon. Again, this is prenatal testing, so before birth. And then testing for infants and newborns. Routine screening within the first few days of the infant's life is done in order to identify CAH. Also, in infants who are female, chromosomal tests are able to be run in order to identify genetic sex. For this particular disorder, genetic testing is typically done within the first 48 hours of life. For this particular disorder, also detection as early on is important in order to prevent infants from dying due to salt-losing congenital adrenal hyperplasia, which leads to hyponatremia, which can lead to death. And then lastly, genetic testing options within young adults and children. Urine and blood tests. These are tests that are done in order to look for specific hormones produced by the adrenal glands to see if they are in range. Also, physical exams looking at blood pressure, heart rate, and symptoms of CAH, which are checked routinely. Also, early detection for this is necessary in order to prevent further progression of the disease. And then the typical cost of testing for any age is $95 to $2,000 for a genetic test. That is the range. This is the end of my presentation. I hope you enjoyed.